the topic, you must have little bit of knowledge of thermodynamics, because all the kinetics of enzymes, basically it is the advanced or little different form of chemical kinetics what we study in +2 and even in graduate courses. So before wasting time, I will be going little fast, so try to cooperate with me because there are plenty of things in the list. So what is enzyme? So enzymes are basically it is protein, okay, and known earlier in the very like in the start of this dimension of biology, they were known as biological catalyst, which is inseparable, assumed to be inseparable from the living system. The Pasteur postulated this hypothesis and said that this biological catalysis process cannot be separated. Later, it was separated by uh, it was later it was seen that the extract of the ferment yeast, even extract of yeast can carry out the fermentation process, and the hypothesis was excluded. So basically this enzyme is biological catalyst, mainly proteins. Nowadays we have little variation in, so it is not only protein, there are several other biomolecules also, which is now becoming to have, now being known to have biological catalytic function. So so it is biological catalyst, basically inorganic molecules we can say, which will include all that. And what does it what it does? So it accelerates the rate of reaction, chemical reactions capable of like it can not utilize in the, it do not get utilized into the process, and it is ready again, it will be ready for the next cycle of catalysis. So it can carry out multiple reactions, okay. So that is the basis for immobilization of enzymes. Now it is industrially various products in green chemistry being produced through this immobilization of enzymes. Yeah, am I audible?
Yes, yes, yes. You're audible.
Okay. Yes, yes. So and basically enzymes follow all the chemical kinetics. So all the reaction kinetics of basic like chemical compounds, it all follows, and uh, the basic other catalyst which is in the chemical lab or chemistry, known in chemistry, basically inorganic molecules like heavy metals and others, which is known as catalyst, and they also enhances the rate of chemical reactions, but the magnitude with which the enzymes enhances the rate of reaction is commendable, and this is one of the the key factor for like evolution of living organism in this planet. So it is very highly specific and provides enough ample opportunity to be regulated for the cellular benefits and even for benefits of mankind. So you can differentiate. There are several similarity in catalyst and enzyme, like both enhances rate of chemical reactions. Both are involved in enhancing the chemical reactions, and both do not disturb the chemical equilibrium and do not get consumed into the process. But it has certain key differences, which is one of these specificity of enzyme is very unique and very high, whereas in catalyst it is not very specific, because the fundamental principle on which catalysis, like it enhances the rate of reaction, is the providing catalytic surface, or it provides ambient condition where the collision of molecules can be more probable and the reaction could happen. But the rate the magnitude with which it enhances the reaction is far less than enzymes, and only the fractions of the enzymatic efficiency it can be like catalyst can perform. So that is why this is the major driving force of nowadays, where the chemistry, the lab chemistry, wherever it is possible, and enzymes is known, protein is known involved, known to be catalyzing those kind of chemical reactions, industrially important chemical reactions, enzymes is being preferred. And that domain of chemical engineering utilizing this potential of enzyme is green chemistry. And another one important difference is the mechanism of regulation, which is very much offered by enzymes, but in catalyst, only temperature and pressure can be regulated, but you have to compromise with the efficiency of the reactions. So that is very crucial difference I wish to share with you. As I mentioned, if you will compare that, you see most uh, अभी तक के, I think most of you know Hindi. Anyone confirm? Most of you know Hindi, right?
Yes, yes.
Yes, sir. So like orotidine monophosphate decarboxylase, okay, OMP decarboxylase, it is one of the, like, this is the most efficient enzyme known to this, in planet. So like you see the dimension, the magnitude with which it enhances the rate of reactions. Can you see you see this is 10 to the power 17, 10 to the power 17 times. If a normal reaction, which is probable to, the probability of getting that catalysis is 1. So you, at the same time, if you have enzyme at that place, it can convert 10 to the power 17 molecule in 1 second. So it is very, like, you cannot imagine. If you physically compare with your productivity, so then you will realize that how efficient these enzymatic systems are. So so apart from this catalytic power, enzymes provides specificity at various level, like bonds. Then some enzymes are very particular about kind of bond it hydrolyzes, like or kind of bond it is it synthesizes, like peptide bond, peptidyl transferase, you might have heard. Group specificity, some are very particular about functional group of that chemical compound. Some are very particular about entire substrate, their structures. Some are very particular about optical chirality of that molecule, geometrical, and even various kind of cofactors specificity are also associated. Basic enzyme structure and cofactor, everybody might be knowing that enzyme is, most of the enzyme, not all the enzyme, but most of the enzymes are protein. And proteins got this this function of catalyzing biochemical reaction in a living system, particularly because it can have various conformation. Okay, it can have various conformation, and particularly the conformation which it acquires in that biological buffers, okay, or biological living system. So that where they have some certain places which can accommodate very specific kind of substrate, and bring out some conformational changes and catalyzes the reactions. So that can be like many enzymes are very simple enzymes which has only protein part, and many have not many, most of them have conjugated kind of like enzymes which has protein and other non-protein portions. And many times non-protein portions can be like settled, like when reaction is being carried out, those substrate will come and support in catalyzing those reactions, and later it get associated and dissociated. So that kind of like substrate kind of support, when it is provided by non-protein part, it is called as co-substrate. And when any inherent inorganic organic part which is very much closely associated with the protein, and without denaturing the protein, that organic component you cannot remove, such kind of a integral organic component is known as prosthetic group. So like these cofactors, these cofactors are like, from enzyme point of view, it is not known when, if you are not into the subject. But from the general perspective, nutritional perspective, we all know that vitamins are very important. Vitamins are very important for our survival. So more, not most, all the vitamins are the part of enzymatic catalysis system. Okay, all the vitamins, they are basically, they are cofactors. They are cofactors, or that co-substrate which comes together with enzyme and catalyzed very specific function in biological system. So for exam point of view, I can tell you that in examination, you will be from PG level to the PhD level, or even services level examination, you will be getting direct questions and very easy to answer questions from this section. So what are the kind of like, I can quote, many times they ask what kind of chemical reactions it is catalyzed by this vitamins or cofactors. Like here you can see, uh, we have listed some of the this one. So just a minute. So we have listed, means I have listed some of the things, like you have thiamine. Thiamine, you might have heard thiamine pyrophosphate. It is integral component of pyruvate decarboxylase, and most of the decarboxylation reaction, wherever decarboxylation reactions you have, you will be having thiamine pyrophosphate, because carbon transfer is involved. Okay, carbon decarboxylation reaction. So similarly, flavinin and NAD, FAD and NAD both are involved in oxidation reaction and dehydrogenation reactions. So these are very important in oxidation reduction chemical reactions. Similarly, pyridoxal phosphate, this pyridoxal phosphate is uh, uh, in glycogen phosphorylase, and most importantly, these are very important in amine transfer, transaminases kind of reaction, wherever, like amino acid, if you will study the metabolism of amino acid, might be Dr. Ashish might have discussed it. Their pyridoxal phosphate, PLP, is very integral component of the reaction mechanism being executed in metabolic pathway of amino acid. So pyridoxal phosphate is integral part of those amine metabolism. Coenzyme A and further coenzyme A and even biotin, coenzyme A and pantothenic acid. Okay, it is not written here. So pantothenic acid, both are very much important fatty acid metabolism, where they catalyzes transfer of acyl group. Similarly, biotin is important transfer of carbon, and 5-deoxyadenosine cobalamin. It is very few reactions, few biological enzymes which needs this cofactor, and one of these methylmalonyl mutase. And many times it has been asked. You can rate it three-star questions. So similarly, tetrahydrofolate, it is also carbon transfer reaction. So similarly, you see kind of reaction which catalyzes, like you have acyl oxidation reduction, clavin, acyl transfer, lipoic acid, it is involved in acyl transfer, then oxidation reduction, again NAD involved, then pyridoxal phosphate transamination reaction, tetrahydro transfer of one carbon, thiamine pyrophosphate aldehyde transfer. So these are vitamin precursors which is involved in the type of kind of reactions it is executing or supporting the enzyme for execution of this kind of reactions. So I hope this is very important, and if I'm telling you, this is not exclusive. When I was a student, these are, I have not, this is not new thing. It is everywhere. So these kind of collections you must have if you are preparing for such examinations. You download those photographs. You download those photographs from Google. Just Google it. You will find several photographs there. Very informative photographs. So just download it and just keep on scrolling. In past time when you browse for Facebook and Insta, so instead of that you can browse the photographs. You make a specific folder so in your phone, or I can suggest you, there is a slideshare.net, one website. Earlier it was in LinkedIn. So that SlideShare, there you will find plenty of PPT slides. So whenever you are free, you are not extensively involved in study, that time you can utilize those kind of activities. Because you see, anybody on this, most of, I can say, not anybody, most of them only, they read 5 or 6 hours sitting on place. Okay, but if you utilize non sitting hours for your enhancement of knowledge, that will be very crucial, and even that will not be painful, and effortlessly you can study, effortlessly you can consume more and more information, and that will help you in, not in short term but in long term. Okay, so this is one simple suggestion for all of you. uh, so again, coming to the topic, so nomenclature of enzyme, it is one of the very like essential, crucial thing, because enzymes were, somebody started it as a naming enzyme as a ferment. Okay, somebody enzyme, like enzyme is, the enzyme name itself comes from, enzyme means nothing but yeast. Okay, and enzyme, n means inside, like inside yeast is the enzymes. Okay, so the name itself have been originated from such. So it was like those who discovered the enzymes, they named their based on their perceptions. So many times it was seen that kind of chemical reaction was used for naming of those enzymes, like transacetylase. Many times even none of type of reaction, or nor the substrate is being specified by name of the enzymes, or the prefix and suffixes like age, in, they also have several contradictory uses. Like you have lactase, where you have lactase means it acts on the lactic acid and hydrolyzes the reaction, whereas the same fumarase we have. Okay, so fumarase we have, so these fumarase is like isomeric kind of reactions. So both are like, it is different. Then this prefix and suffixes have been has ill used. So similarly, protein, for it is in use in most of the protein enzymes. So it was felt that we must have a uniform nomenclature, because the increasing use of enzyme, it is very much important for development of biological science. So there was in 1964 a committee was formed, a commission was formed for nomenclature of, and formulating a guideline for nomenclature of these enzymes. So in 1964, they published one draft in which they classified enzyme into 6 classes, and those 6 classes were given a unique name, unique digit. Okay, 6 classes given the unique digits, and each enzyme were given the four-digit enzyme commission number. And this enzyme commission number is very unique, and like everywhere it is used. You use whatever names, but you have to mention the although there is a standard guideline for using the name in standard literatures, but you can use trivial names also, but you must mention the EC number, because it will unique, it is unique number, and it will see, designate that specific enzymes. So that nowadays the development of bioinformatics has been very enormously supporting the science. So we have one database called Explore Engine. So this database is maintained by IUBMB. Earlier, MB was not there, only International Union for Biochemistry was there, but now it is MB, International Union of Biochemistry and Molecular Biology. So this IUBMB nomenclature, they maintain a database called Explore Eng, where you can visit for all your informations related to nomenclature. So traditionally, enzymes were classified into 6 classes: oxidoreductase for oxidation reduction reaction, transferases for transfer of any atom group. Okay, we discussed about carbon transfer, aldehyde transfer, amine transfer, acyl transfer, aryl transfer. So those kind of reactions you will be, it comes under transferases. Similarly, hydrolysis, it is one of the largest, class of enzymes where you have, all hydrolysis kind of reactions, like any reaction which ends with the production of water or uses water as, nucleophile. So that is called as hydrolysis. So this is also very, all the hydrolytic digestive enzymes, you might have trypsin, catalase, diastase, any maltase, any kind of those digestive enzymes, they comes under hydrolysis. Okay. So fourth class is lyases. Lyases, it is very confusing with ligase. Okay, it is very confusing with ligase. Lyases are basically, they are, they are, any group of when like any group is removed from a bigger molecules, like if a molecule has other functional group, and in a reaction it is desired that a specific functional group must be removed, or any portion of that molecule is removed. So that removal, if it is catalyzed by not involving water, then it comes under lyases. Okay, and so it comes under lyases. And next class is isomerases. We will go through this later. So isomerases means it will be any enzymatic conversion, either for geometrical transformation or for chiral transformations. Those enzymes comes under isomerases. Then ligases. So ligases is the synthetic joining of two molecules. Like you might have heard of ligases in molecular biology. T4 DNA ligases, so which joins nucleotide molecules or two DNA molecules. So basically they form phosphodiester bonds. Similarly, other kind of bonds which is joined, means they are joined, like carbon carbon or phosphate phosphate sulfur carbon bond. So these kind of they are called as ligases. Okay, the synthesis of bond. So basically this EC number has 4 components. First one is the major class, which is here since it is 1, so first class is oxidoreductase. So second number is a subclass, and subclass means what kind of what is the subclass, means what kind of category, means under oxidoreductase which category of this enzyme is. So like in oxidoreductase, hydrogen and electron donation, electron donor or electron acceptance is one of the criteria of categorizing those enzymes. So here one is the hydrogen or electron donor molecule. If it is alcohol, then it is like 1. Okay. So similarly, the third one is the hydrogen or electron acceptor molecule. So if it is NAD and NAP, it is 1. Okay. So and 27 is the entry number. It is very very everywhere. You might get questions from this. The fourth number, first number now everybody know. So fourth number is the indicate, like in this category, when it was entered. So this the 27 is the lactate dehydrogenase, and this 27 is lactate dehydrogenase, and the entry of that lactate dehydrogenase into this category was at 26th number. So this 26th number, so that is why 27 is there. So I just take you to the Explore Enzyme database. So here you see this is the website where you have enzyme by class. So this enzyme by class, you go, you have EC1. Okay, EC1 oxidoreductase class. So EC 1.1, the CHOH alcohol group transfer. Then 1.2, it is aldehyde ketone transfer. Similarly 3, CHCH transfer. Similarly, you have plenty of like 99 things which is being transferred. So again, if you go to a further inside, like in CHOH group donors, you have acceptor. Which one is the acceptor? So NAD plus NAD plus is acceptor. So this acceptor has several entry, you can see. So first one is alcohol dehydrogenase. If enzyme name was 1111, so this is alcohol dehydrogenase. It is asked in the exam several times, alcohol dehydrogenase. So similarly, alcohol dehydrogenase, if NADP plus is the donor, means acceptor. Similarly, you have plenty of enzymes, list of enzymes which is named. And I'm telling you, nowadays, it was earlier it was considered that memorizing these kind of enzymes EC number is not, means should not be asked in exam. But nowadays I'm telling you, these are being asked in the examination. Not very rare enzymes, but the enzymes which is of frequent use, like so those frequently used enzyme, you must be aware of their EC number, at least the category for that. So transferases, you have second class transferases. You have 2.1 is the one carbon group transfer, 2.2 is the aldehyde or ketone group transfer, 3 is the acyl transfer, glycosyl transfer. Similarly, you have several classes. If you go into the detail, the one is the methyl transferases. Again, in methyl transferase, you will have hundreds of entries. Okay, so similarly, this kind of like you can get into details of, if it is any exam, if any you want to know about any specific enzyme EC number, you can visit the website and you can directly search by entering the EC number. So similarly, here also hydrolases. So most of the hydrolases, they act on ester bond. Okay, ester bond, or they can act on the glucose glycosylases. Okay, they also can act on a ether bond, peptide bond, carbon nitrogen bond, acid anhydride bond, carbon carbon bond, halide bond. So it's a very big class of enzyme where you will have you will have various entries. Okay, so similarly, this is how, like you have several example of hydrolases. So not to, so in liases, this cleaves reactions like CCCO, the same kind of reaction, same kind of hydrolysis what we discussed in hydrolysis. So here also we break the bonds either of CS, CC, CS, or any kind of bond, CO. Here also we break the bond, but only difference is we are not involving any water molecule here. Water molecule is not involved. Okay, so you have carboxylases, aldehyde liases, oxylases. One of the like pentose phosphate pathway, if anyone have studied HMP pathway or pentose phosphate pathway, there you have these kind of enzymes. The use of these aldoses and aldolase and ketolase kind of enzymes. So those are liases. Oh, I hope I'm audible and I'm doing justice. Okay, am I audible, sir? Anyone please confirm? Yes. Hello.
Yes, sir, you are audible.
Okay. So another is isomerases. Next class is isomerases. Fifth class of classification is isomerases. So here again, racemases and epimerases. You have in racemases and epimerases several enzymes. You can see all racemases and then epimerases and all. So basically, these are the enzymes. They are involved in isomerism, epimerism. You might have heard of epimer of glucose. and racemases, the change in the optical rotation D2L and other kind of isomerases. So there are several cis and trans isomerases are there. Intermolecular oxidoreductases are there. Like you many times you have condensation, self-condensation reactions. There you will go through intermolecular rearrangement, intramolecular rearrangement. So there also these kind of enzymes are involved. So if any specific enzymes you wish to check the EC number, you can visit the website, you will get. So ligases are basically opposite of what we do in hydrolysases and lyases. Okay. So in lyases, what we do, we hydrolyze it. Means in lyases, what we do, we break CC bond, CS bond, CO bond, and CN bond without utilizing water. But here ligases, they synthesizes these kind of bonds, CC, CS, CO, and CN, and these bonds are, these syntheses are very much like several example of ligases are there in being used in molecular biology, where carbon oxygen bonds are being synthesized. Even you have like cycloligases, and even penicillin. In penicillin also acylases are there. So these are classical example of ligases. So another class you will not like everywhere, it is not in the textbook, still it has not got the space in textbook, most of the textbook. And but a seventh class of enzyme, E7, okay, enzyme commission 7, has been given to translocases. Okay, translocases means anything which can translocate. Okay, any protein which is involved in translocation of compound. Okay, so in membrane you might have seen that these are the hydrophobic barrier. These are the hydrophobic barrier. Okay, these are the hydrophobic barrier here. Okay, so the hydrophilic molecules are very difficult to cross this biological membrane. Okay, so there came the role of protein, and many proteins, they are like flipase, flopase, you might have heard. So those kind of proteins which are involved in translocating these kind of molecules, they are given a separate class in the enzyme classification, and they comes under E7. So this, if they are translocating only hadrones, hadrones means H plus, or so those kind of transporters, like all the ATPases or P type H plus, these transporters, they comes under translocases. So don't get surprised if any EC number starts with 7. And even I have not come across with any questions, because it has not has got any place in textbook, that even questions are not there, but they are not far when you will be getting questions from this class of translocases also. So movement of molecules or ion across membrane on their separation within membrane. So this is a tweet of IUAMB, where they have declared in 2018 that they have given a new enzyme class, translocases, apart from the previously reported 6 classes. Okay, so this is very important, and you just try to visit this IUBMD website. There are very beautiful like informative videos are there, animated videos are there, the many very like difficult concepts which are very difficult to comprehend. It might help you there. So visit the IUBMD website. This organization has trends in biochemical science and biofactor. These 2 important journals released by this IUBMD, and another newsletter which is regularly released each month. So you can subscribe through the mail. If you are interested, you can you will be updated, because there are various confusions. I will give you just homework. Okay, you might have heard about synthases and synthetases. Okay, synthases and synthetases. Many times even a numbering of that it will do. So such kind of confusions are being addressed by this IBMB, and so you, if you will have the recent knowledge, you will become pro. Okay, pro in your area. So if you wish, you can be updated. So coming to the second dimension of this topic is the reaction kinetics. Okay, reaction kinetics, how enzymes catalyzes chemical reaction. So basically what enzyme does. So enzyme basically enhances the rate of chemical reactions. Earlier reported catalyst, inorganic catalyst, basically what they does, they provide a surface. Okay, surface. Through on that surface, various substrate molecule have their sub binding. Okay, some affinity. By affinity to those surfaces, since surfaces has affinity to those surfaces, their movement is little seized. Okay, movement is little seized, and there is a probability that two substrate can collide with each other and get enough energy to form the product. Okay, so that is how catalysts work. So and both enzymes and catalysts, they do not disturb the chemical equilibrium, but only they enhances the rate with which it reaches the equilibrium. Okay, so a chemical like a enzyme catalyzed reactions can be depicted as when an enzyme combined with substrate, like they form enzyme substrate complex. So enzyme substrate complex, again it will undergo some transformation, it will form enzyme product complex, and later then little bit of conformational change will release the product, and that conformational change will be required to restore the activity of enzyme. Okay, so if you see the typical chemical reaction from thermodynamics perspective, so what happens, why a chemical reaction happens. Okay, so anything in this planet happens for stability. Okay, anything on this planet, any chemical reaction or anything in this planet happens for stability. Okay, so the substrate wants to get more stable because energy is less. Okay, energy is less. So since product has less energy than substrate, it wants to go there. Okay, substrate want to become product. So how it will become product? So it cannot directly go to they can lose, substrate can lose the energy and become P. If it is so, then substrate cannot exist, because if it is so, then substrate will not be stable. Okay, so to have its identity and the product's identity, they are separated by a energy barrier. Okay, although they can get more stable, but only when they will cross this energy barrier. Okay, so this energy barrier for conversion of substrate into product is known as the activation energy, is known as the activation energy. Okay, and this active, to acquire this activation energy from here to here, the substrate need to need to gain that energy. How that can gain? So basically that activation energy will be gained by the environment. Okay, by the environment. So the how the environment will help in gaining that energy? So by when substrate in a environment, they are exposed to temperature, pressure, kind of various chemical interactions. So these chemical interactions lead to the gain in the energy. gain in the energy of the substrate, and it becomes it get enough threshold activation energy to reach at the threshold energy level, from where the probability of transferring getting into conversion to product is equally probable. Okay, so this transition state where this it can be converted to substrate and product is equally probable. So this activation energy is very much required. A very famous once, like it was in a class, a teacher teaches many students. Okay, a teacher teaches many students, same content, same methodology to all the class students, but why all do not become successful, or means not successful, means all do not perform well in examination? Why? The reason being the activation energy, the energy barrier. If any student has energy barrier is very high, so that information, it will not be sufficient. That energy what teacher is providing is not sufficient to reach at that threshold level and become the product. Okay, so practically also, so if anybody having previous knowledge, anybody having prior knowledge, or anybody having basic knowledge, so they have better, they have the activation, the requirement of activation energy is very less. Okay, so even a little support of the teacher, they can that can take the student to cross this barrier. Okay, to cross this barrier, and this barrier, from there it can be easily, so always in your life also try to overcome this barrier. So this is the activation energy barrier. And in life also, don't try to just pass the exam, don't try to become IAS officer, don't try to become anybody. Only thing you try to become is just raise your activation energy. Just raise your internal energy, so that it is easier for you to cross this barrier. Okay, so this is the analogical example from your life. Okay, so the difference between substrate and product is this is the difference of energy. Okay, which gives stability to the product, and this is the actual difference of energy between those substrate and product, and it is basically called as Gibbs energy, free energy. Okay, Gibbs free energy. And the conversion, the difference is called as Gibbs free energy difference between. So I hope okay, yes, this is reaction coordinate diagram. So it is, although like it will not be asked in the exam, but so many times if anybody asks that have you gone through the reaction coordinate diagram of this enzyme, then it will go out of mind. What is reaction coordinate diagram? So reaction coordinate diagram is nothing but the progression of enzymes activity. Okay, progression of enzymes catalysis with respect to free energy. Okay, with respect to free energy level. Okay, so that is known as what reaction coordinate diagram. Okay, so this reaction coordinate diagram we will be using like now onwards in many slides. Okay, so I thought I given too much time on this slide. So anyway, transition state, another thing, like at the threshold activation energy, you will have a transition state. Okay, a state of yourself where you can understand everything, where you can gain information very fastly. Okay, where you can gain information very fast and can transform yourself to the product. So you see, once upon a time I was I was like reading 5 pages. It was very difficult. Reading 5 pages was very difficult for me, and it took I think hours to sit and comprehend those pages. But now, because I have raised the energy level, okay, I have raised the internal energy level, that comprehensive and reached at this transition state, where I can read daily, on the daily basis, 50 pages. If I read, even it is not painful for me. Okay, so this is how I'm telling you in so this is how the transformation also happens. A substrate, uh, even small weak interactions brings their energy level up and reduces the activation energy barrier, and a time reaches in the transformation process in the reaction coordinate, depicted in the reaction coordinate, where at that transition state the substrate can easily get transformed into product. Okay, so here, when enzyme is used as a catalytic medium, it reduces the energy barrier to very, you can drastically reduces this energy barrier, and drastic reduction in energy barrier can bring out this substrate into product, and this process becomes very easier. So it was uncatalyzed energy difference, and this is the catalyzed energy difference. Okay, so how these transformations happen? So what happens when any assume a reactions? Okay, so if you are catalyzing in a container, you are wish to catalyze the reactions. So you have substrate, you have all the liquids, all the buffers and other things in the place. You have all the substrate. So what happens when enzyme go there? Okay, enzymes go there. So enzymes start interacting with the substrate. Okay, start interacting with the substrate. So interaction is basically what? Interaction is basically these type of interactions is non-covalent interaction, very small non-covalent interactions. And these small non-covalent interactions really, through that equivalent interaction, in non-covalent interactions, they gain the energy. Okay, and then they gain internal energy and reduces the barrier. Again, this enzyme substrate conformation, this enzyme substrate complex, they at the active site, they bring out some that catalytic process, or there also they undergo into various kind of covalent interactions and metal catalysis and other kind of things. So those changes those substrate into product, and later conformational changes in the enzyme releases the product. So similarly, the same it is mentioned here. Okay, so enzyme basically they do not affect the reaction equilibria, but they enhances the rate of reaction so that the system can reach the equilibria at very rapid rate. Okay, so the same thing it is written here, and many times if there are multiple steps involved in the catalytic process, then the process which has highest energy barrier, okay, which has highest energy barrier will determine the formation of product. Okay, so basically there may be 10 steps involved, but if a step whose barrier activation energy barrier is very high, then that transformation will determine the rate of reactions. So those kind of reactions where in a multi-step process, or basically we see it in a pathway. So those that specific reaction is called as rate limiting reactions. Okay, rate limiting reactions. So these things we discussed. So for like from chemistry point of view, previous class also we have discussed in pH. Like you can law of mass action, through law of mass action you can reached to this level, when any reaction at equilibrium, when substrate is converting to product, then at equilibrium, this law of mass action, through law of mass action, we can determine this equilibrium constant. Okay, equilibrium constant. So K equilibrium will be the product upon substrate. And since there the stoichiometric coefficient is 1, so it is not written. Okay. Similarly, when this K equilibrium, it is dependent on many factors. I told you that this K equilibrium was inherent property of that chemical transformations. Okay, that chemical reactions. So that inherent property is determined by the nature of substrate. Okay, one thing. Another thing, it is also impacted by environment. So in previous classes I discussed with environment means pressure and temperature, both will be determined this the equilibrium constant. Another thing, it can be correlated with free energy. Okay, free energy. So if you have little bit of exposure for school days in chemical, and you might be having knowledge about it, then del G, like free energy, can be related with minus RT ln Q. Here K is the equilibrium constant, R is the rate constant, T is the temperature, and this is very means everywhere it is used, and many times a small numerical. Okay, where they will ln they will just modify it in the multiples of 10, so calculation becomes easier, and they will ask you these questions even in objective also. Many times it is being asked. Okay, so rate of any reaction is determined by the concentration of reactant and by the rate constant. Okay, so similar the same principle in the enzyme also. So velocity of any reaction, here rate is given as V. Okay, velocity of any reaction is determinant of a rate constant K and the substrate. Okay, so substrate, like if there is a single substrate, okay, if there is a single substrate, S is converted into P, then it is a first order reaction. Okay, first order reaction. So you might be aware about the order of any reaction. So if there is one substrate, then it will be one order reaction. If there are two substrate, okay, and their stoichiometric coefficient is 1:1, then it will be of second order of reactions. Okay, so that will, like, we will not get into the complexity of the order of reactions. So here we will be like we will be considering it to the single order reaction, because multiple orders, although in biological system you will find very less reactions where you will be having single substrate and getting transformed into product. Most of the time we encounter with multiple substrate reactions, but for simplicity of this transformations, chemical this mathematical calculations and all, for comprehensions, we will be limiting it to the first order reactions. Okay, so if it is of second order, like there are two substrate S1 and S2, the same velocity will be like law of mass action you use, and you will get the velocity of reactions. And you just another thing to just add, the unit of reactions you can calculate. Okay, the unit of first order reaction is second inverse per second, and the unit of second order reaction is mole per mole per second. Okay, so similarly in this transition state, further transition state, basically any substrate reaches a transition state mainly because of non-covalent interactions between the substrate, enzymes, and to the environment in which it is catalyzed. So it is very important. And earlier it was induced, it was it was told that lock and key hypothesis was given, where it was told that already the enzyme has a definite shape kind of pocket is there, we call active site. So that pocket is already fixed, it's permanent in shape. So only the specific substrate can fit into that pocket, and the enzyme catalysis happens. So this is the inherent property of enzyme, and this one substrate, it is catalyzing. Okay, so this enzyme substrate complex. So but this theory, this hypothesis was not appropriate, although there is a concept of active site. Active site is there, but it is not permanent, it is not fixed, because it was observed that many enzymes, they have same shape. Okay, same shape, but they are not being catalyzed by the catalyzed by the same enzyme. Okay, and it contrary also, it was seen that many times enzyme have such shapes, but it is not means being catalyzed. So this lock and key hypothesis has not gained much attention, and later it was you might have heard about induced fit model. Induced fit model. So for lock and key hypothesis, it was suggested that this is classical example of lock and key hypothesis, where dihydrofolate reductase, where active site is already fixed. Okay, and already fixed, where this tetrahydrofolate comes, binds, and NADP comes and bind, and they carry out the that transferase reactions. Okay, oxidoreductase reactions. So this is the hypothesis on, and that enzyme support this lock and key model, but it was felt that this lock and key model is not supportive, because there is a in various modeling, now we can model the protein, we can have the complete structure, three-dimensional structure of protein. So it was seen that there is a active site, although there is active site, but the size and the dimensions of active sites is not constant. Okay, it changes with times. And it was also seen that many times a different molecules having different molecules, like they have their certain differences in the structure, but they fit into the active site and that inhibit the enzymatic reactions. So there are several counterproductive things against the lock and key model was observed. So there is a modern notion of enzymatic catalysis. It was proposed, a group of scientists, they come with the modern notion of enzyme catalysis, in which it was said that enzyme must be complementary, as said in the this lock and key model, but this complementarity should not be with the subject, with the substrate, but with the transition state. Okay, transition state. So how this transition state, how substrate reaches to transition state, I discussed about. There are several weak interactions. Okay, these weak interactions of non-reacting parts. Okay, several things happens, like non-reactive part, like if you consider an enzyme, there is an active site. Active site is where you will have various functional group which will be involved in the catalysis process. But why enzyme is so big? Enzyme is so big? Why? Only catalytic site could have done the purpose. So the rationality of having an enzyme of this big nature, and so the reason is, first, it protects the active site, and second is it contributes enormously in the catalysis process. Okay, they might not be directly involved in catalysis, but they are providing energy payment. Okay, energy payment means they interact with the substrate enough interaction with the substrate makes the substrate energy rich. Okay, energy rich means it raises the internal energy of substrate so that it can easily reach the transition state. Okay, and after transition state, the catalysis or whatever the process at the active site happens, and the product is formed. So the modern notion is this is complementary, and complementarity is to the transition state, not with the substrate. Okay, so this is also like it gives the rate enhancement. This is how the rate is enhanced. Okay, this also gives why enzymes is highly specific. Okay, you might have, like we have discussed about bond specificity, stereo specificity, and other kind of various specificity we discussed. So this specificity, earlier it was assumed in the lock and key hypothesis that this specificity is basically because of the type of active site and the shape of active site. But here I am discussing, here I am telling that this binding energy determines that which substrate reaches the transition level early. Okay, I means that the which substrate will have more interaction with the enzyme and ultimately reach to the transition state and become complementary to the active site and undergo the process of catalysis. So these interactions is the key crucial thing which will bring out this substrate to the transition level. Okay, so if any substrate, they might be having the similar kind of structure. Okay, in lock and key hypothesis, they might be having similar kind of structure, but if it is not interacting enough with the enzyme and not reaching the transition state not reaching the transition state, it will not be catalyzed, it will be out of the catalytic process. Okay, so this energy payment and this enzyme substrate interactions prior to the transition state is very crucial, not only for catalysis but also for selective specificity. I hope I am I am able to convince, I am able to convey what I mean to. So how these binding helps? So this first thing is entropy reduction. When enzyme interact with the substrate, it restricts the relative motion of the substrate. It restricts the relative motion of the substrate. You consider the same two substrate of same conformation but not having proper interactions. Okay, if it is not interacting means enough entropy level will not be reduced, although they are having same shape, but enough entropy reduction will not be there. So the collision probability of the substrate will be lesser. Collision probability of the substrate will be lesser, but if they interact sufficiently, entropy reduction will be sufficient, then their probability of collision and reaching to the transition state become much much easier. So another thing is desolvation. What happens if you just assume the hydrocolloids? Okay, like enzyme, most of the enzymes in the means works in the biological system, and most of the things are means water hydrophilic. So in a hydrophilic situations, you consider that most of the portion, like enzymatic portions, that is hydrophilic. Okay, they are water bonded. Water is bonded all around the enzyme, this one. So this desolvation process desolvate the enzymes, desolvate the enzyme, which makes the substrate which makes an opportunity for the substrate to enter into the enzyme process. And this removal of water also provides additional energy for reaching the transition state. Further, there are several non-covalent interactions which generate favorable energy, And if any substrate, any two substrate having all these, they will be catalyzed with the same enzyme. If two substrate with the same shape and size fitting to the active site but do not possess all these will not be catalyzed. I hope you all got what I'm trying to tell you. So here it is very beautifully this demonstrated, illustrated, that you see if you have no enzymes, so like it is very much like it is reaching at this place will be difficult, like lot of energy will be required. Okay, so this is the uncatalyzed reactions. Okay, so when in a lock key model, when you have enzyme, this enzyme name is stickase something, they just hypothetical enzyme they so stickase. So stickase is the enzyme which trying to break this stick. Okay, so this enzyme, if it is fitting with the substrate, okay, if it is fitting with the substrate, then substrate is too much, like substrate is still rigid. Okay, substrate is still rigid, and although they might convert into product, but the rate at which this conversion will happen, it will be much lower, it will be much lower, right? So in the case of induced fit model, what happens? Although the substrate, this substrate is not, complementary to the active site, because this active site is complementary to the transition state. So what happens? This substrate, which is not complementary to the active site, it start interacting with the enzyme, and this interaction brings out conformational change in the substrate. Okay, so that conformational change, how this conformational change has brought about? This conformational change is brought about by these kind of interactions. Okay, these kind of interaction, and these kind of interactions brought about these conformational changes, making this stick complementary to the active site. Okay, active site, and this active site, and makes this stick fit into the active site, where active site the catalytic process happens. Okay, catalytic process happen. And how this catalytic process happen? This catalytic process happen with the conformational change. Okay, so this model of this lock and key model is not sufficient to explain the high rate of reactions, but this induced conformation or modern notion of enzyme catalysis is sufficiently sufficiently suffice the reasons, rationality of the enzyme catalysis. I hope everything is clear. Okay, any problem till now? Any questions till now? Hello.
Hello, participant. Please ask any doubt regarding this topic. So you can clear your doubts. So this is important topic from a Department of Biochemistry student. Ravani?
I will be happy to answer if any query. Okay, no issue. If you do not have problems, means you understood everything.
Akshata? Miss Akshata Kare?
Yes, sir.
Is there any problem?
No, sir, no.
Okay, okay.
Okay. So now we will enter into the another dimension of like when enzyme substrate, like enzyme substrate is substrate has attained the transition state. okay, through non-covalent interactions. Now it is at the active site, okay. When it is an active site, there are various kind of catalysis process happening at the active site. That is the part of this portion. So basically, although there are like they might classify into another category also, but the majority of the catalysis falls into these three classes, okay. So that is basically first is general acid base catalysis, okay, general acid base catalysis, and the covalent catalysis and metal ion catalysis. General acid base catalysis, we will go one by one here, okay. Sorry for this one, just a minute, keep, okay, just a minute, back here, okay. Okay, yes. So what happens in general acid base catalysis, like as name suggests, that there will be either acid or base will be involved. Either acid or base will be involved means, in the acid base concept we have seen that there is a pair. If a portion is acid, then there, if it releases H minus, it will become base, okay, conjugate base. So what happens here, this in general acid base catalysis, even water or even weak acid, okay, weak acid, they can donate H plus, okay, they can donate H plus, and after donating H plus, they will stabilize the substrate, okay, and they will gain enough nucleophilic potency to donate this electron to the the electrophilic moiety and bring out the catalysis process, okay. So basically this acid base type of reactions are very much common, and you might have studied it since class 9th, okay. So this specific acid base catalysis is the acid base catalysis where you have water molecule, okay, where you have water molecule as an acid or hydroxyl ion as a base, okay, then it is called as acid base catalysis. Many times what happens in biological system, these acid and bases are water, and if water is not there, there are several amino acid moieties which has the potential to act as acid and base, that we have discussed earlier, right? So this potency of various amino acid has been crucial in catalyzing various processes, various enzymatic reactions. So most of the, like all the digestive enzymes, what you might be knowing, all the digestive enzymes like protein digestive enzymes, carbohydrate hydrolysis, all those enzymes are basically uses this acid base catalysis process. And if in many cases, if it is not like not only water, that will be supported by either in the the buffer can make any, like the buffering capacity or the pH of buffer can make water as a acid or base. Similarly, other moieties also can act as a acid or base. So in the environment also it decides, like and like the same moiety, if it is in different pH buffer, so they might react differently, they might act differently, okay. So if it is water, then it is specific acid base catalysis. If it is general any other than water, that may be either weak acid or weak bases, or even the moieties from amino acids or other biomolecules, they are considered into general acid base catalysis, okay. So the various amino acid group has been crucial in this acid-base catalysis, like you have glutamate and aspartate. So these are proton donor, like weak acid, and then lysine and arginine. They can also, act as a acid and base. Then cysteine, then histidine, then serine and tyrosine. So in different, microenvironment of the active site, which determines the nature of these active amino acid residue, whether it will be acting as a base or acid. Okay, many times what happens that this hydrogen donation capacity in normal buffer, it cannot be done, but at the active site this hydrogen will be taken by some other amino acid. That other amino acid will be acting as a base, will take this H plus, and this CO minus CO minus will become a nucleophilic site, which can donate and catalyze the reactions. So it depends on the environment also. Okay, so active site and environment in buffer and active site is entirely different. So just keep in mind. So that covalent catalysis, this is also one of the not means it is common type of catalysis mechanism, and it should not be seen as an isolation, because many times what happens, even a single enzyme uses all the catalysis method of catalysis, because there is at active site, even a metal ions can be the part of active site, even a covalent catalysis can be the part of active site, same active site, and even acid-base catalysis can be demonstrated the same active site. So it should not be seen as in isolation, but all three are the methods through which the catalysis process is brought about. So this covalent catalysis, what happens here, you will have covalent bond formations, and here in acid-base catalysis, it is ionized bond formations little bit. Okay, and the later covalent bond formation. So here permanent covalent bond is formed, and later those bonds will be broken, and the moiety will be released. So these kind of covalent catalysis will be there. So again, metal ion catalysis, most of the oxidation reduction reactions, they are supported by metal ion catalysis, and even many the dehydrogenation is also oxidation reduction type of reaction, and many other reactions also can be using these metal ions. So metal ions, this what basically does, so metal ions basically it stabilizes the substrate, or it stabilizes the substrate ions, and even these metal ions can act as an electron acceptor. It can act as a electron acceptor. If you see that magnesium ion, we have seen that in the CS lecture, that even ions also can, in Bronsted concept, ions also can act as a acid, because it can accept the lone pair, so this metal ion can accept the lone pair, and that, and this is assignment that you just note out, just make out that kind of metal ions. Okay, and the major enzymes to which it is associated. Okay, so there are two kind of metal involving, there are two types, with two types metals can be involved in catalysis process. One is in the co-substrate form. Co-substrate form is whenever catalysis is being performed, then metal ion will be coming and will be assisting in the catalysis process, and again will dissociate from the enzymes. So that metal ion will not be the part of enzymes. Okay, but in many times these metal ions are the integral part of the enzymes, and where you cannot remove that enzymes into means you cannot see that enzyme in isolation, and that enzymatic activity will not be there if you will remove that enzyme and that metal ion. So such cluster, you might have heard about FES clusters, like in electron transport chain, various complexes are there. So in those complexes, you might have heard about FES cluster. Okay, FeS, iron sulfur clusters. So those iron sulfur clusters are embedded into the proteins, and you cannot remove that prosthetic group without denaturing that proteins. So metal ion, because those metal ions are inherent part of their catalytic mechanism, and without those metal ions, the entire process cannot be done. So metal ions also, and how it performs, basically it stabilizes the process, stabilizes the substrate ions, and can act as a acid. So now we will see the enzyme kinetics. Okay, so enzyme kinetics is there is a separate means domain of enzymology, where rate of reactions, how the changes in response of a substrate or changes in the experimental parameters can affect the rate of reactions or rate of catalysis of an enzyme is being studied. And this most of the whatever enzymology we are studying, basically why we are studying, one thing is to generate information. Okay, we generate information about the enzyme. If we know the information about the enzyme, like we are, there is a term called active site mapping. Okay, so active site mapping, what means, this is part of enzymology, where we try to map the active site. Okay, we try to know the dimensions of active site. And why we are knowing? Because after knowing the dimension of active site, we can modulate that enzymes in our favor. Like if we know the, like all the herbicide, insecticide, and other things, these are the like inhibitors. Okay, inhibitors of enzyme. So what they do, so how it has been discovered, or how it has been made, because the enough information about any enzymes, enough information about their moieties, enough information about their three-dimensional structure and their active site, allow us to design a molecule which can have the efficient interaction with the enzyme and reach the transition state, and permanently block the enzymes, and those inhibition we can achieve. So that kind of inhibition will be favorable or desirable in any counterproductive enzymes. So this has, so similarly, one is the information, second is the utility applications. So both in both areas, enzyme kinetics is crucial. So you might have heard about this Michaelis-Menten equation, Michaelis-Menten equation. So basically, derivation, it is like anybody, when I was UG, it was little difficult for me that how derivations, and very like it was seen that, oh, Michaelis-Menten equation derivation, you can derive it. Okay, yes. So although it is not very difficult, just you focus on how the steps, how it has been done. So basically, the hypothesis of Michaelis-Menten was the steady state kinetics. Okay, steady state kinetics. So they assumed that in pre-steady state enzymes, we are considering this enzyme substrate, makes enzyme substrate complex, and this enzyme substrate complex will become into enzyme and product. It releases the enzyme and product will be released. So this reaction, they ignored this initial portion, yani ki pre-steady state. So pre-steady state is the momentary time where the enzyme and substrate are not in, like not in enough proportions. Okay, not in enough proportions. And ideally it was considered that when first molecule of ES complex is formed or product is formed, so that the time when reaction started and the first product is formed is basically seen as a pre-steady state, and This pre-steady state is basically you cannot even you cannot even not observe pre-steady state, but theoretically pre-steady state is there, and when pre-steady state is over, you will have all in sufficient amount, all in saturating amount. Like there you will be having enzymes, you will be having substrate, you will be having enzyme plus substrate. So, and you will be having E plus enzyme plus product also. So that steady state it was assumed, and C or both worked out to have a correlation between the velocity of enzyme and the substrate of enzyme, velocity of enzyme, maximum velocity of enzyme, and the substrate of enzyme. So basically in this equation you have V naught. V naught is the initial velocity. V naught is the initial velocity, and later just a minute. You have initial velocity, then Vmax is the maximum velocity which enzyme can attain. S is the substrate concentration, and Km is the Michaelis constant. Okay, so all are measured. These all parameters can be measured experimentally. Okay, these parameters can be measured experimentally. So if a new enzyme is there, you can find out, you can substrate concentration you can know, initial velocity you can know, and maximum velocity you can know. So you can easily find out what is the Km. Okay, and Km is the inherent properties of that enzyme. Km is the constant, Michaelis constant, and it will be the inherent properties of that enzyme at that particular condition. Condition means that will be temperature, pressures, buffers, pH, and other things. So at that particular condition. So now we will go into little bit of derivation how it was done. So you see, this is the central part. Okay, ES, enzyme substrate complex formation, is the central part. Okay, so this is two process. Okay, enzyme and substrate get converted into ES. So ES is the product for these reactions. Okay, and ES is the forward reaction, and ES is the substrate for the reverse reaction. Okay, so rate of ES formation means we are talking about forward reaction whose rate of constant is K1. So what we can do, we can law of mass action we can use the K1 into enzymes involved in this conversion. Means how much enzyme is there? Some enzyme will be free enzyme, and some enzyme will be in the complex form. There are only two forms of enzymes are here in entire sequence. So one is free enzymes, either here and here. Okay, another one is in association with the substrate. So if we consider the total enzyme, E total, okay, if we consider E total, so E total will be what? E total will be E free enzymes plus enzymes into S. Okay, enzymes into complexation. Okay, so if we consider this, if you wish to have like this enzymes. Okay, this enzymes means enzyme total minus ES, enzyme total minus ES, the concentration of ES. So here the rate of ES formation will be K1 into S into E, and instead of E we can write ET minus ES. Okay, so this is okay. So second rate is dissociation, how ES is lost. Okay, how ES is lost. So ES is lost with two reaction. One is the backward reaction of the previous, this one. Okay, and another one is this formation of product. Okay, so rate of ES breakdown is the result of two reactions. One is this backward reaction, so K inverse, K reverse into ES. Okay, only one substrate, and for this reaction ES is the substrate. Okay, so K2 into ES. Okay, so you consider this equation first and second. So this is the crucial thing. If you get this is very easy. Okay, so rate of ES and ES formation and ES breakdown you can work out, and then you assume at steady state, we assume that rate of formation of ES will be equal to the rate of formation of breakdown. Okay, so ES will be in like in sufficiently saturated condition. So if you assume that condition, so these two equation will become rate, since rate is equal, so these two equation 1 and 2 will become equal. So you just equate this equation and solve for ES. Okay, K2, K1 is the constant. Okay, two variables are there. One is substrate. Okay, and another is the ES. Okay, so just solve for the ES. Okay, solve for the ES. You will come across. This equation. ET into S divided by substrate concentration plus K inverse plus K2 by K1. K inverse is the reverse rate constant. K1 is the forward. K2 is the forward second forward reaction. Then divided by K1. So if we make this entire K1 is also constant, K2 is also constant, and K reverse is also constant. This constant, if we assume this is another constant named as Km. This is Michaelis constant. What is Km? Km is again, this can be, Km is again, this is K inverse, okay, K inverse plus K2, okay, K2 divided by K1, okay. So you see that K inverse means rate of the rate constant of the reaction, which is breaking this ES. Okay, upper is the rate constant which is breaking the ES formation, divided by the rate constant which is forming there. Okay, so this becomes Km. Okay, Km. This is called Michaelis-Menten constant. So if you just simply put this constant into Km, you will come with the enzyme E total into substrate divided by Km plus S. Okay, Km plus yes. And simply enzyme substrate, here you come here. When you have maximum, when you have here you can see initial velocity of any reaction will be K2 into ES. Okay, velocity of any reaction will depend on how enzyme substrate will be releasing the product. Okay, like we consider that enzyme substrate formation is one part, and enzyme substrate, converting those enzyme substrate into product is another part. This portion, okay, this portion. So for for initial velocity, initial condition, this reaction is the determining constant for the velocity. Okay, if you have faster in a saturated condition, you just consider the steady state kinetics, where the rate, how it will be going forward will determine this rate of reaction. So V naught will be K2 into ES. If we solve this, put this value of ES, here, you will come across with this K2 into ET into S divided by Km plus S. Okay, and K2 into ET, we can consider it as when enzyme becomes total enzyme, then it will be maximum velocity. Okay, so this is how this Michaelis-Menten equation have been worked out. And what is the benefit of having this Michaelis-Menten equation, and why people are so fond of this equation is, I can tell you that most of the enzymatic reactions follow this. Most of the enzymes, they follow this Michaelis-Menten equation, and this equation is the basic application of what we have studied in the reaction kinetics in our chemistry classes at graduation and pre-graduation level. So and this equation allow us to interchangeably determines the if you have, if you know the substrate of substrate, how much substrate you are putting. If you know the nature of the enzyme Km, okay, velocity, maximum velocity, you can easily get the velocity of reactions. So intercurrent, we can, if you know the 3 parameter, one you can easily determine, means 2 parameter, one can easily be determined alternatively. Okay. Further, this enzyme also gives insight where you can, if you will plot this uh, substrate concentration and velocity, you will get a rectangular hyperbola. Okay. So here you see that initially you will have, very little increase in substrate concentration, you will gain more in velocity. Okay, but later in section, even uh, in large increase in substrate concentration, you will gain very little increase in. So it means in the later portion you will get saturation curve. Okay, velocity will ideally reach at the V maximum. Okay, and initially this means initially the rate of gaining velocity will be much higher. Okay, so how, by this equation also we can reach at this level, this conclusion, by you assume that you assume that that you have maximum substrate. Okay, substrate is very high. Okay, when substrate is very high, okay, so you can easily two number, one is very low and one is very high, you can easily consider this without having much impact on of this number. Like if you have 20 crore rupees, and if 20 rupees will go, it will not have impact on your financial thing, right? So similarly here if you have big amount of substrate, large concentration of substrate, then Km will not be an issue. Okay, so simply V naught will become V max, V naught will become V max. So at large substrate concentration, you will ignore Km and the V will become V max. This is the ideal hypothetical situation. So similarly, if in the initial condition, if you see, when substrate concentration is very low, then what you will do, again you substrate concentration, since it is very low, it means Km is very high. So this substrate you can easily leave, and you can easily reach at the other level. So when Km, and in a specific condition, when Km become equal to substrate concentration, velocity becomes half mark. Velocity become half mark. So these notions has been used widely for the catalytic to measure the enzyme kinetics of comparing the different enzymes and the kinetic parameters. And one more thing I wish to add, that even same enzyme can have different Km. Okay, same enzyme can have different Km value for different substrate. Okay, because this Km value defines the on the basis of substrate level. Okay, so may have a enzyme may have may require some substrate in low concentration, some substrate in high concentration. So Km value differs. Okay, it is based on the substrate. So again, what happens in the previous slide we have discussed, you see, this is K inverse. K inverse means this reverse directions. Okay, this is reverse directions, and K2 is this one. Okay, so rate of ES expenditure by rate of earning, ES earning, is the ratio. Okay, so if you consider, like Km is also like traditionally being used as the it also determines that which substrate, to which substrate it has more affinity. Okay, like if you the you can measure, like if you consider that this enzyme has this kind of shape of active site, so this can catalyze this substrate also. You compare the km value, so definitely basis on the km value of different substrate, you can preferably decide that this enzyme gives preference to which substrate. I hope you all are getting what I'm trying to say. So if you know the km of, you see, you know the like, you just considered analogically like this, like you have one enzyme, and one enzyme have the potency to catalyze 5 similar substrate. Okay, have the potency to catalyze 5 similar substrate. Okay, how these 5 substrates are similar? Because these 5 substrates have means they can have means they have similarity to the active site. Okay, so they can all can be catalyzed. So when you will measure the reaction rate in the experimental condition in laboratory, you will find that there is a contrasting difference in all these 5 this one, all these 5 similar molecules. So basis of km, on the basis of km of each substrate, you can decide that which substrate is more preferential for this enzyme, which substrate is more preferential of this enzyme. This was traditionally used. Okay, this was traditionally used, but it has several problems, and one of the problem is, like, any substrate, any enzyme has more affinity for a substrate. Okay, it is decide here. Okay, it is decide here, means it will be decided by k1. Okay, so if you consider the km very carefully, you just listen, I seek your attention. You see, so if you see the km, this is the reaction of, this is the constant of 3 rate constant. Okay, so one is k1, another one is k reverse, and k2. Okay, so if ideally S is how much affinity S has for E, this enzyme, it is determined by K1 rate constant. Okay, by this rate only we can say that this substrate has the affinity for this enzyme, but not by other, because since there are 2 other equations, so if a Km is low, it doesn't mean that this K1 is means high. This may be because K inverse and K2 is also low. Okay, so Km, it was the relevance of Km was questioned by various stalwart, and later it was modified. So further adding to the kinetics for traditionally, where now we have lot of, like, earlier on paper we cannot extrapolate this graph. Okay, this graph we cannot extrapolate, and we need to have more number of entries, a record point at various level. So it was very difficult to extrapolate like all the data from this graph. So for a easy conversion, we just extrapolated that curve, that hyperbola curve, into a linear curve, and that graph is famously known as Lineweaver-Burk equation. Okay, Lineweaver-Burk equation. So what, and several kind of simplified equations were given, and that Lineweaver-Burk was given. It was accepted too much, and now also it is being used in laboratory, and in gemmology classes also you might have come across with some practical on this session. So how this Lineweaver-Burk equation was came into existence? Just simply, whatever the Michaelis equation was there, the reciprocal of the Michaelis-Menten equation, you just simplify, you will get this simple equation of Lineweaver-Burke plot. And if you see the Lineweaver-Burke plot, Km and Vmax for an enzyme is constant, Km is enzyme is constant. Similarly, 1 by Vmax is constant here. So for a linear curve, you might have heard, you might have been knowing that y is equal to mx plus c. Okay, mx plus c. So c is the intercept, okay, and m is the slope, okay, and y and x are the variables. Okay, so similarly here are three variables. 1 by v naught is one variable, 1 by s is another variable, and 1 by v max is another variable, and 1 by mx is another variable. Means not variable, means it is constant. So this 1 by v max is the intercept. Okay, 1 by v max is the intercept, and km by v max is the slope. Okay, so if you plot this 1 by v versus 1 by s, okay, velocity, inverse of velocity by inverse of substrate, you will get a linear curve. Okay, you will get a linear curve, and this linear curve you will have a slope. Okay, slope is basically, slope is defined by tan theta also. Tan theta, okay, tan theta will be like, you take any this one, okay, you take any this one, this is theta, okay, theta. So this will be tan theta by 1 by v naught, 1 by v naught, okay, divided by this one, 1 by s. Yes, okay. So this tan theta will lead to the this will give you the slope. Okay. So the slope is km by v max, km by v max here. In this case, km by v max, and this point is the 1 by v max, 1 by v max, and this point is the minus 1 by km, since it comes into the fourth quadrant. Third quadrant. So this is slope km by v max, and this is the substrate concentration on this one. So in practical, in laboratory condition, you can determine that v naught you can determine, you can determine v naught, you can determine v max. Okay, you have the substrate concentration, so you can easily find out the km value, and in interchangeably, if you have other things also, you can find others. Okay, so turnover number, the km, the lacuna what we referred in the km, that km cannot determine the affinity of an enzyme to any specific substrate. So for that reason, kcat or turnover number is being used. Okay, turnover number is nothing but it is the extrapolation from the rate limiting steps. Okay, rate limiting reactions. So in the rate limiting reactions means that reaction determines the speed of entire catalytic process. Okay, so if we consider a multi-step process, if this K3 we consider as a rate limiting reactions, so basically this rate constant will become crucial, and this will only determine that rate of the entire process. Okay, rate of the reactions. So what happens for a maximum velocity, if you know the kcat, they denoted as a kcat. Kcat means that crucial rate limiting step and the rate constant. Okay, if you have kcat and total enzyme multiplied, you will get Vmax. Okay, right? Okay, so this is for maximum velocity. You must have kcat and enzyme total. So this becomes first order reaction. This becomes first order reaction and can be easily you can calculate. But what is problem in the in the practical scenario? You will not get single step you will not get single step enzymatic process, and
problem is that many times, it is our assumption that this process, out of 5 process, which one is rate limiting, okay, which one is means. So we need to find out which one is rate limiting, and for measuring that rate limiting step, you must have not only theoretical insight but also practical insight into those entire processes. So it becomes very difficult, but for numerical solving problem and other things, you can use this KCAT, this one. But if you know the KCAT, it is called as since it is first order constant, so the notion is per second, okay, per second. So simply by definition, you can call it the number of substrate molecule converted to product in a given unit of time on a single enzyme molecule is called as KCAT, or turnover number, okay. And turnover number, if you consider for many biologically known like enzyme, you can see catalase. Catalase has the highest turnover number, okay. Catalase has the highest turnover number. Similarly, carbonic anhydrase, okay. So so this is further, many enzymes are of many substrate, okay. So those enzymes are these are more practical, but the algebraic expressions, or to reaching that mathematical model for measuring different rate constant and the enzyme kinetic parameters, it is very difficult, okay. So these multimodal enzymes, although they are Dalziel and Albergi, they have tried to do that, but here this is out of scope of discussion, and you will not be getting any significant questions from those areas. So it is not of means not worthy to discuss here, but we can discuss the multi substrate reactions and how it is how reaction is catalyzed. So many these type of reactions, basically they involve the group transfer reaction from one group transfer to other group, kind of reactions, oxidation reactions. So what happens, these kind of reactions, they fall into two category. One is ternary complex formation and no ternary complex formation. Ternary complex formation means if enzyme substrate, if any enzyme encountering two substrate together at one moment, where enzyme has bounded with two substrate simultaneously, is called as ternary complex, okay. And in such enzymes, those that complex formation is very crucial, because they will be transferring one moiety of one substrate to other moiety, okay. So for transferring those moiety, both substrates should be bounded to the enzyme, okay. So in that case, this ternary complex formation is very essential. And considering that it can be of two types, one is ordered mechanism, another is random order mechanism. So in ordered order, what happens, the two substrate what you have, that must appear in a order, sequential order. So why sequential order is required? The reason being, hypothetically, reason being the first substrate which binds to the enzyme, it brings out some conformational changes in the enzymes, okay. And that conformational changes is very much essential for binding of other enzymes or other substrate, okay. So without inducing that conformational changes in the enzyme, second substrate cannot bound. So the binding of first is the crucial event, then only second will bind, and further the enzyme catalysis is there. So this kind of reaction is called as ordered reaction, and mostly the group transfer reactions you will find this kind of encounter with it. Another one is random order reactions, okay. So in random order reactions, as name suggests, that there is no, like, both ternary complex is formed, but the sequence is not determined. Okay, so one can bind first and other can bind, vice versa. Okay, so this is not necessary that S1 should bind first, because there is no crucial active site is there. Active site is really means uh, fully operational, and that active site can accommodate any of the enzymes without any restrictions on the order. So that kind of reactions are order displacement. And further, to have these kind of for depiction of these kind of reactions, there is a nomenclature given by Cleland. They have given random these are just denotation what we have seen here. Okay, so ping pong click and mechanism, and then these are denotation mechanism that you can learn. Okay, so now this is enzyme inhibition. Although I know it is very it became too lengthy to discuss all this, but anyway, we are another 20 minutes. I will try to bind it up. I do not have much experience of speaking for this much time, but I am also taking extra effort, and I will request your kind patience for another 20 minutes. Okay, so enzyme inhibition is very applied field of this this enzymology, and human existence has been appreciating, and as days progressing, its existence is very crucial. Many of the detergent nowadays what we are using, earlier they were simply simply without any enzymes, but now whatever detergent we are getting, it's little costlier and have all those enzymes which can degrades various kind of stain forming chemicals used in our households. So this is how many enzymes has been of commercial importance, and their regulation also becomes very important, particularly in agriculture, in herbivores and herbicide and pesticides development. So basically, inhibitors are the enzymes of reversible or irreversible inhibition. The name suggests reversible inhibition means the inhibition which can be reversed. Okay, means it do not denature or do not make enzyme permanently non-utilizable. Okay, like enzyme can regain their potency of catalysis once the appropriate condition is provided. But irreversible inhibition is the counterproductive, and once it binds, it do not means it completely brings out permanent changes in the enzyme structure or conformation, and it renders enzyme unutilizable. Okay, irreversible inhibitions, whatever the inhibitors we will be studying, all will be out of scope to irreversible inhibitions. Okay, this is very crucial. I am telling you, many times reversible and irreversible, I have did the mistake many times. Which of the following is irreversible inhibition, they will ask, and they will give all the competitive, non-competitive, and one will be none of the above. So this has been mistakenly comes into our conscious mind that these are the irreversible inhibitions category. So be just get it that all are reversible inhibitions, and basically we call it any organic molecule which can modify, enhance, or retard the velocity of enzyme is called as modifiers, And if it is positive modifiers, we actually call as activator. We call it activator. And if it is a negative modifier and retarded reactions, it is called as inhibitors. Okay. So enzyme inhibitions basically any molecules which interfere with the catalysis and retarding the process of enzymatic reaction is the enzyme inhibitors. Or how these inhibitions is brought about is either that enzyme can we will not discuss about permanent damage, but in reversible manner also, either that inhibitor can bind to other portion of non-catalytic portion of enzyme, non-catalytic of portion of enzyme, and brings out some conformational changes, which is not conducive for catalysis of the main enzymatic pathway. Okay, so that inhibitors can bind to other surfaces, other pocketed, and also even at the active site also it can bind. Okay, so many inhibitors which has similarity with the substrate. Okay, many inhibitors which has similarity with the substrate, they can directly approach the active site, and in active site they will sit for means they will compete for the active site, and they will make physiologically enzymes unavailable for the catalysis of the reactions. Another condition is they will bind to the other non-catalytic portion of the enzymes, and they will make this entire bring out some conformational changes, and they will make the entire catalysis process non-conducive. Okay, so such enzyme inhibitions, particularly analog substrate analog type of inhibition, it has been widely utilized for development of neochemical molecules for herbicide and pesticide applications, and even medicines also. So one of the, like aspirin is the acetyl salicylate, inhibit the enzyme involved in the prostaglandin synthesis. So aspirin is basically painkiller, and prostaglandin is the pain inducers. Okay, so all the painkillers you might be getting, so that painkiller is the non-steroid anti-inflammatory drugs, NADs. Okay, so those NADs are the aspirin family drugs, and those are the pain relievers. Okay, there another kind of drugs which can relieve the pain is the steroidal drugs. Okay, so steroidal drugs, but steroidal drugs have several complications, so aspirin is widely used. So several molecules of these pain relievers is in the market, and basically all are little modifications of others. So there are these all are we discussed. So reversible inhibition is basically categorized into three classes: competitive inhibitions, where we have substrate analog, the inhibitor is similar to the substrate; uncompetitive inhibitions, where that substrate binds with the enzyme other than active site. They can bind with the enzyme inhibitor, enzyme substrate complex, and even yes, enzyme substrate complex, and this one. So because that even enzyme inhibitor complex can have the opening for the substrate binding, but the efficiency becomes lower. So second is having, and last one is non-competitive inhibition. Basically we call it mixed type of inhibition, in which you will find competitive and uncompetitive mode both, but in a special cases we can assume this non-competitive inhibition also. So basically what we discussed about reversible and irreversible reactions. So in that we can add some many a times this many times these inhibitions are very crucial for regulation of metabolic enzymes, and in our enzymes in our metabolic pathway, several of the product of some other pathway try to regulate the enzymes of one pathway. So means because those product, this is how interconnected our metabolic pathways are. So reversible inhibition and even irreversible inhibitions are also being utilized by the utilized as a regulatory mechanism in the metabolic regulations. And one of the irreversible method is covalent modifications. Okay, covalent modifications. You might have heard about in DELA protein. Okay, DELA protein. You might heard about counterproductive proteins. Just blocking or degrading those proteins will activate the regular pathway. So that kind of covalent regulations are also used in metabolic system. And so further, basically those enzymes which can be regulated, those are very crucial. So now those things we have discussed in this one. So coming to the competitive inhibitors are the substrate analog type of inhibitors which can bind to the substrate, because they have usually the analogy, a shape analogy or the functional group analogy with the actual substrate of the enzymes. Okay, and this can bind with the inhibitors directly compete for binding to the active site of the enzyme. Okay, so if you take this this reaction representation, this enzyme substrate forms enzyme substrate complex and enzyme product formation in regular cases. But what happens, basically enzymes amount will be decreased if inhibitors will bind and make it unutilizable for product formation. So basically enzyme is becoming enzyme is competitive for both the pathway. So if you have substrate, more of substrate here, so this pathway will go in this direction. And if you have more of I, so then this pathway will go in more in this direction. So this is the regulation of equilibrium. So if you have equilibrium biased toward ES formation, then reaction will go in this direction. And if you have equilibrium more of I, then it will go in this direction. So what happens, so this is the Michaelis-Menten equation for this inhibition. So what happens, Km become alpha Km. So what happens, alpha Km means what? So the Km requires, like, to reach the half maximum velocity, you will have more amount of substrate is required. Km is, this alpha Km is apparent Km. So the Michaelis-Menten constant under inhibitions. Okay, so that is apparent Km. So apparent Km is nothing but the under inhibition, what is the half maximum velocity. So if you have this alpha Km, you consider this under competitive inhibitions, what happens, this Km becomes alpha times. So to reach the same half maximum velocity, you will have more amount of substrate is required. But if you have enough substrate, still you have the potential to reach the maximum velocity. So Vmax is not changing, but the Km will change. Okay, so bound inhibitor does not inactivate the enzyme. When inhibitor dissociates, substrate can bind and react. Okay, or when substrate concentration exceeds the inhibitor concentration, then definitely you can attain the maximum velocity. And this is very common type of inhibitors and very easy to comprehend. And the same, if you will take the reciprocal plot of this Michaelis-Menten plot, so you will get a Lineweaver plot where your slope will be like alpha times. Okay, earlier slope for a reaction was Km by Vmax, but now slope will be a Km by Vmax. And since Km is increasing, okay, since Km is increasing, the slope will increase. If you have more more potential competitive inhibitors, slope will be more upside than slope will be slope of the less potential inhibitions. Okay, so here you can see this is the alpha 1, then alpha 2, alpha 3. Means if you have potential inhibitions, the graph will be in this line will be on upward side. Many times these kind of graph will be given and you will be asked for this one. So this is alpha Km by Vmax. So just simply, logically also, if you will see that if basically what happens, if I is there, basically some of the enzyme will not be available for the reactions. Okay, some of the enzyme will not be available for the reaction. So in order to reach the, definitely you will be not getting the same reaction rate. So in order to reach the maximum velocity, you have to increase the substrate concentration simply. So there are several example, and one of the example is this dihydrofolate reductase, this methoxate. So this methoxate is the inhibitor of dihydrofolate reductase, and several other succinate dehydrogenase inhibitors is malonate and other. So in uncompetitive inhibitors, what happens? This is the like E and S can form enzyme substrate complex. Okay, enzyme substrate complex they can form, and enzyme substrate complex will bind to the I. Okay, will bind to the inhibitor, and what it does, it siphon of the ES complex. Okay, from this siphon of the ES complex. Siphon of the actually, so siphon of the ES complex. So the E plus S brings out ES, and ES get converted. So basically ES complex is gone. So Vmax, so you will not get maximum velocity. So this maximum velocity, this Michaelis-Menten equation in this case will be like Vmax S by Km plus alpha S, and this alpha is the basical loss in the this is another constant of inhibitor constant we say. So this inhibitor constant will be based on this complex. If you define the Ki, use the law of mass action. So ESI will be the ES plus I. So ESI by product divided by ESI. So you have Ki. So inhibitor effectively what it does, it removes some of the fraction of enzyme molecule, and I binds to S. So S needed to reach the half Vmax, again it will decline. Km decline by the same amount. So similarly, what you can see here, again Km required for, again here you see this Michaelis-Menten equation. If you will consider, then here you can see this Km by Vmax remains constant, means slope, you will not have change in the slope. Km by Vmax remains constant, but Vmax is under problem. So half by Vmax is the 1 by alpha times the natural Vmax. Okay, so Vmax keep on reducing if you have this one. Okay, so half Vmax will go on. So So if you have more potential uncompetitive inhibitors, this point will be keep on rising, okay, because this is 1 by Vmax, okay. So ultimately, this you will be compromising with the maximum velocity, okay. So mixed type of inhibitor, although these mixed type of inhibitors in in context, most of the enzymes are multi-substrate enzymes, so you will be not getting this one, but the sake of theoretical understanding, mixed inhibitors is any enzymatic inhibitors which can bind with both, either directly to the open enzyme or to the enzyme substrate complex, making it totally totally unavailable for forward reaction, or not even if it is you consider it an equilibrium thing also, then also this, since it can react here, so entire ES complex and E both will be siphoned up from the main process, and you will be compromising with the rate of rate of reactions, okay. So what happens when you will see the Michaelis-Menten equation? In this case, K becomes alpha times and S becomes alpha dash. So both Km and substrate both will be compromised. Both will be compromised. So in this case also, you will see that velocity in if velocity, you will velocity also it becomes alpha times alpha dash times, and this Km becomes alpha times. Since there are two inhibitors which has different potency to bind with enzyme, single enzyme, or enzyme substrate complex. So both the potency of the same inhibitor will be different, okay. So alpha and alpha dash will is here, which has different value, okay. So the velocity will be means the decrease in velocity and increase in Km will be not with the same magnitude, okay, will be not with the same magnitude. So if it is with the same magnitude, if it is with the same magnitude, we will reach an ideal condition where this alpha for both will be same, and that condition we call it non-competitive inhibition, okay. But this is ideal condition, and we will rarely get in actual scenario. Considering this Lineweaver burst plot, if you see, then here you see Km will be alpha times, so slope will be increasing, okay, slope will be increasing, and Vmax will be alpha times, okay. So Vmax also will keep on increasing, okay, Vmax will also keep on increasing. And since this intercept you consider, so this intercept will also will become if you will see the intercept, since Km here at the y-axis, x-axis, so x-axis at the Km also, it also keep on increasing. So this is how in mixed inhibitors you will have changes in both Km and S, Km and maximum velocity, but with which magnitude both changes they differs. And in one single condition where both we consider both enzyme substrate and enzyme combines with the same affinity with the inhibitors, then this is an non-competitive inhibitions. Again, these are the same thing what we discussed. So some of the example, like you have competitive competitive inhibition example, succinic dehydrogenase. So substrate is succinate, and inhibitors you have glutarate, malonate, and oxalate, okay. Similarly, you have very rare example in uncompetitive inhibitors, very rare example. And non-competitive inhibitions, you will have various metal ion requiring enzymes creating like EDTA and all. You might have in any extraction procedure we use EDTA. any extraction procedure, we use EDTA or EGTA just because to chelate the divalent cations and make the enzyme extraction in proper conformations. So this is all about today, and actually there are a lot of topics which ideally should get a place, but since time was very less and it was very difficult